Hello friends, in this video we are going to discuss about bacterial plasmids. So first of all, what are plasmids? Plasmids are naturally occurring small circular extrachromosomal autonomously replicating fragments of DNA present in prokaryotic and few eukaryotic organisms. Extrachromosomal means these are present in addition to bacterial genome. Autonomously replicating, that is, these are self-replicating. These have their own origin of replication. Plasmid term was coined by Joshua Lederberg in 1952. These are not required for bacterium survival. That is, these are not vital for bacteria but carry genes that provide various advantages to plasmid host cell. It means that cell in which plasmids are present have advantage over cell in which plasmid is not present. Example, antibiotic resistance. If the plasmid present in the host cell has genes for antibiotic resistance, then this host cell is resistant to antibiotic. And the cell in which this plasmid is not present, these are not resistant to antibiotic, that is, they are killed in the presence of antibiotic. Now the facts and terms related to plasmids. First is size. Plasmids are much smaller than bacterial genome. These carry additional genes. These carry the genes in addition to the vital genes that are present in bacterial genome. These exhibit wide range of size varying from 1 to over 400 kilobase pair. Some are as large as 2 megabase pair. These are known as megaplasmids. Second is shape. Plasmids have 5 different types of shapes. First, nicked open circular. DNA has one strand cut. This type of the plasmid is double-stranded circular and its one strand has cut. Relaxed circular. DNA is fully intact. So it is double-stranded circular and it is fully intact. Linear DNA has free ends. Supercoiled, completely intact but has a twist, hence smaller. This supercoiling we have already discussed in separate video. You can refer that video. The link is given in the description box. Supercoiled denatured. So these are also supercoiled but some part is lost due to denaturation. Hence, these are little smaller in size. Third is copy number. Copy number is the average number of molecules of a given plasmid per bacterial chromosome. And copy number ranges from 1 to 3 copies to 100 copies. Host range. Number of bacterial species in which typical plasmid can exist. Most plasmids have narrow host range. That is, these are present in limited number of species. Some have broad host range. It means these are present in large number of bacterial species. Fifth, compatibility of plasmids. That is, ability to coexist in the same bacterial cell. So if 2 or more plasmids exist in the same bacterial cell, then these are known as compatible to each other. But if they cannot coexist in the same bacterial cell, then these are known as incompatible. Now let us see properties and characteristics of plasmids. First is extrachromosomal. So these are present in addition to the bacterial chromosome or bacterial genome. These are mostly double-stranded except linear ones. These replicate independently and hence these are known as replicon. Replicate independently means these are autonomously replicating or self-replicating within a suitable host. These are non-essential. That is, these are not vital for the survival of bacteria, but their presence is beneficial and may provide selection advantage, example as antibiotic resistance. So if there are certain bacterial cells which have the plasmid carrying the genes for antibiotic resistance and certain bacterial cells which do not have this plasmid. So in the absence of antibiotic, their survival will have no difference. But in the presence of antibiotic, the plasmid carrying cell has advantage over the cell in which plasmid is absent. That is, those having the plasmid which has genes for the antibiotic resistance can survive, but those in which the plasmids are absent, these are sensitive to antibiotics and hence these are killed. Plasmids are also known as sex factors, conjugates, extra chromosomal replicons or transfer factors. This we will understand better in horizontal gene transfer video. If we talk about size of the plasmid, their size varies. It has wide range of size which ranges from 1 to 400 kilobase pair and in some cases around 2 megabase pair. So their size varies from mini plasmids to megaplasmids. Copy number. The copy number range from 1 to several hundreds. Larger plasmids have fewer copy number, but smaller plasmids have higher copy number. These are generally circular, but linear plasmids exists and require specialized replication mechanisms. So mostly these are circular, but in some cases linear plasmids also exist and these require some special replication mechanism which is different from the circular plasmid replication mechanism. Those with low copy number have partition systems to distribute copies to daughter cell. Some can insert into host chromosome known as episome. This we will discuss in detail in next part of this video. There are certain plasmids which have no effect on phenotype of host cell. These are known as cryptic plasmid. Now let us see role of plasmids. Gene products encoded by plasmids are rarely essential for survival. So the gene products or the proteins which plasmids encode, these are non-essential for survival or these are not vital for survival but provide advantages to the organism such as utilization of unusual carbon sources. So these help in the utilization of certain unusual carbon sources like toluene, etc. Then resistance to heavy metals and antibiotics. Sometimes these provide resistance to heavy metals and antibiotics, and also synthesis of toxins and proteins that allow successful infection to higher organisms. Now the use of plasmids. Plasmids serves as useful genetic tools in molecular biology and genetics. Particularly, these are used in gene cloning, recombinant protein manufacturing and gene therapy research. These can be utilized to introduce, manipulate or delete specific genes within a host cell. So these points we will discuss in detail in recombinant DNA technology video. Now let us see general structure or elements of plasmid as vector. Plasmid vectors helps in cloning small DNA fragments in bacterial cells. And the elements of plasmid as vector as shown in the diagram are origin of replication, antibiotic resistance gene, selectable marker, promoter, 5' primer site, restriction site, inserted gene, restriction site and 3' primer site. Ideal plasmid vector has these properties: small size, high copy number, origin of replication, restriction sites and selectable markers. In order to use natural plasmids as cloning vector, several modifications are required. Hence what we can say that plasmids serve as essential genetic tool in molecular biology and genetic engineering. And these can be used to introduce, manipulate or delete specific genes within a host cell. These we will discuss in detail in recombinant DNA technology video. This is all for today's video. In the next part of this video, we will discuss about types of plasmids. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.